Greetings dear viewer and welcome to Elimu TV where you exclusively watch and learn. I am your teacher Madam Christine Okoth, taking you today through the 9th lesson of Biology Form 1. We're still on the same topic, cell, and today our subtopic is going to be on magnification. Welcome. So this is a preview of what you're going to learn today in this lesson. So firstly, we'll start by reviewing the previous lesson. Then we are going to review the assignments that I gave you in the previous lesson. Then today we're going to learn about calculating the magnification. We're going to learn the rules that will govern us when we're trying to draw anything biological. Then lastly, I'm going to give you an assignment. Starting with the previous assignment. So the assignment was, from the video watched, list down some points you need to consider while handling a microscope. Last lesson we were talking about the microscope and the parts that are assembled to make a microscope and the functions of the microscope. So the video was also on the microscope and how to handle a microscope. So I'll just list a few points that will help us to know what we are doing when we are handling a microscope. So the first thing we have to bear in mind, you should hold the microscope with two hands. Then one hand should be placed under the base and the other one on the arm so that the microscope does not fall and it's stable on your hand. Then another thing that you have to consider while handling a light microscope, always put the microscope in the table somewhere at the center and very far from the edge. Then something else we need to consider, you should use your coarse adjustment knob to raise the objective lens either 1 to 2.5 centimeters away from the edge so that you can see your specimen clearly. Then you should never use your microscope in direct sunlight because this can direct your eyes. You can damage your eyes. Then another thing that you have to consider, if you're not using the sunlight, you're introducing your own source of lighting system, make sure that you put that lighting system 20 centimeters away from the light microscope. So these are just a few of the things that you have to consider while you are handling a microscope. So to the business of the day on magnification. So we defined magnification in the last lesson and we just said magnifying, just from the word to magnify, it means to enlarge something. So magnification is entirely to enlarge an object. So if you are enlarging an object and you're using the microscope, and you're viewing it, so the specimen is first magnified by the objective lens when you put the specimen on the slide. So it has to be magnified by two lenses. First one, the objective lens, and the other one that will magnify is the eyepiece or the ocular lens. That is where you place your eyes. So if you want to calculate the total magnification, it will be those two lenses and you multiply them. So it will be the eyepiece or the ocular lens, you multiply by the objective lens. So that is the formula you have to use when you are trying to get the total magnification. So let us look at some examples, calculations on magnification. So as we've said, if you want to calculate magnification, you'll first start by multiplying the ocular lens or the eyepiece. That is where you place your eyes. For example, in our illustration, the ocular lens here is times 10. It means that it has been magnified 10 times. Then the objective lens is 100 times. So if you want to calculate the total magnification, it will be the ocular times the objective. That is 10 times 100. That will give you a total of 1000. So the specimen has been magnified 1000 times. So let us look at more example on our table. So the first set of example, the eyepiece lens is times 20, then the objective lens. So let us look at more examples on our table. The first set, the eyepiece lens is times 20 and the objective lens is times 8. So if you were to get the magnification, we'll just multiply the 20 and the 8. That's simple mathematics and you get 160. And remember to put the times sign. So that one means that the specimen has been magnified 160 times. The second example, you have an eyepiece lens of times 14, then you have an objective lens times 10. So you do the maths, 14 times 10, you get 140. So it means this specimen has been magnified 140 times. The third set of example, we have the eyepiece lens of 25 and the objective lens of times 40. So the total magnification will multiply the eyepiece 25 times 40. It will give us times 1000. Then the last set of example, we have an eyepiece lens of 19 multiplied by the objective lens of 25, and this will give us a total magnification of 475. So still looking at magnification, so let us now look at biological drawings. So sometimes you are given a specimen in a lab or in an exam, and you are required to draw this specimen on a piece of paper that you have been given. So how do you go about drawing this diagram? There are some rules that you have to adhere to while you are drawing this diagram. So let us just look at some of those rules. So the first one, and the obvious thing, while you are drawing, you need to have a sharp pencil and a rubber, and you also need to have a very clean, plain piece of paper. Then before you start your drawing, make sure you draw the outline first of where you're going to draw your diagram from. Then do not draw a structure before you have identified it. If you have been given a specimen, make sure you know what that specimen is before you start drawing the specific specimen. So the next thing that you have to consider, you should make large drawings and leave enough space for heading and labeling. If you have given a piece of paper, make sure you use that piece of paper well. So make sure that your drawing is large enough to be seen and not very small also. Then you should align your drawing in the center. You're given a piece of paper, make sure that your drawing is aligned to the center. Then when you are labeling your diagram, very important thing, remember when you are labeling, do not cross the labeling lines. So in our illustration here, we have two biological drawings. The first one, if you look at that diagram, the diagram has been aligned to the center, the labeling is very correct. Then you look at the other set of diagram. You can see the diagram is very small, you cannot even be able to read the labeling. Then when this person was labeling, he did a crossing which you are not supposed to do while you are labeling. So make sure you remember those points. Then while you're still drawing, your drawing should be clear with fine lines and simple outline. Do not repeat your lines. Make sure it is just overflowing. Then avoid shading the diagram. Look at our illustration here. We have a very beautiful flower. But the mistake is this person was shading the flower. So if you are doing a biological drawing, make sure you don't shade. Just do it the way it is. There should be no shading of the diagram. Then while you are labeling in pencil, keep the words in horizontal, and remember we say that there should be no crossing of labeling when you are labeling the signs. Then another important aspect, make sure you do not use your arrows. Do not use arrows to label the diagram. Just use straight line. And then another thing, remember that your drawing must have a title. For example, you are drawing the structure of a leaf. Just label there the external structure of a leaf. So this is an example of biological drawing. Let us see if some rules have been observed here. So maybe one thing which is missing is the title. So there's no title here, but there's good use of the space. The space has been properly used. There's clear and very strong lines that have been used when they are labelings, and the labels are very clearly written. Then there is also a scale bar which is very appropriate. The lines touch the label. The lines should be touching the label structure. For example, you are illustrating or you are showing the flagellum. That flagellum, from the flagellum to where the name flagellum has been written, it is touching the labeled structure. Then there is no any shading, there is no crossing of labeling. So this is an example of a good biological drawing. So we have been we have been calculating on magnification when you are viewing something from a microscope, from a light microscope. Now, when you've been given a structure to draw, you have the actual structure, then what you'll be drawing on the piece of paper, that is the image of that specimen. Now, how do you go about calculating the magnification of that thing that you have drawn? So we have this equation, this formula. If you are calculating magnification, that is based on a biological drawing, it will be the image divided by the actual size of the specimen. Now, sometimes you are told to calculate the actual size. How do you calculate the actual size? You will do the image. The image is what you've drawn on the piece of paper. You divide by the magnification. Sometimes you're just given the actual size and the magnification. So how do you go about it? You just multiply the actual size multiplied by the magnification. That structure there is trying to show us how to calculate the formula. For example, the actual size will be the image divided by magnification. If you are calculating the magnification, it will be the image size divided by the actual size. So let us do one example. So we have this example. Joydell drew a leaf on a piece of sheet. The actual length of the leaf was 4 centimeters and the image length was 10 centimeters. So what is the magnification of the leaf? So start by identifying what do you need to calculate, what have you been given there. So he was drawing a piece of sheet and the actual length is 4 centimeters. That is, the specimen was 4 centimeters. Then what this person was drawing on the diagram, the biological drawing on the piece of paper, that is what we call the image, because it's a reflection of the actual specimen. So you have the image size as 10 centimeters, then you have the actual size as 4 centimeters. So how do you calculate the magnification? It will be the image size divided by the actual size from our triangle there. Then so magnification will be 10 centimeters divided by 4 centimeters. So we do that mathematics. The units will cancel out, the centimeter and the centimeters. Then 10 divided by 4 will be 2.5. So you remember, you should always include the times sign to show that magnification has been magnified. The specimen has been magnified 2.5 times. So that marks the end of our lesson today, and I'll leave you with this assignment. Question number 1. An object has been magnified 275 times. The objective lens is times 25. So you calculate the eyepiece lens. The next question, calculate the actual size of a specimen. The specimen has been magnified 72 times and the image size is 12 centimeters. So thank you very much for watching. If you have any questions, any feedback, any clarification on the lesson or any other lesson, you can hit our Facebook page Elimu TV. Our Twitter handle is at Elimu_KE and our SMS number is 22518. I have been your teacher, Madam Christine Okoth, and thank you for watching. Keep it Elimu TV.